Hello guys, I am Shubham from Tutorial. This video is all about the lipid. So before we start, subscribe to our channel and press the bell icon to get notified whenever new videos out. This is absolutely free from your side, but it will help us grow. So basically lipids are fatty acids, right? Fatty acids, okay? Fats or oil come under lipids. So the things that are important for biology that we will be studying in this particular section. So I'm trying to make it more simple, right? Fatty acids are water insoluble. Okay, water में soluble नहीं होता. Hydrocarbons, right? Chain of hydrocarbon. What is hydrocarbon? Containing carbon and hydrogen. Used for cellular energy storage, right? So fatty acids are highly reduced and thus provide a rich source of stored chemical energy for cells. Okay, this is the energy reserve, right? Storage of hydrophobic fats as triglycerol is also highly efficient because water is not needed to hydrate the stored fats. Okay, water is not needed. So water insoluble is hydrophobic. We can also say lipophilic. Water hydrophobic and lipophilic, lipid loving. Next point, membrane lipids are composed of hydrophobic tails attached to polar head groups. Okay, we have seen this in the membrane lipids. Right, membrane lipids हमने cell biology में discuss किया है. I will attach that video in the I button. Okay, so this is the head, which is a polar, and this is a tail. So the arrangement is like this. Okay, cellular membranes are composed of variety of lipids. There are many lipids, including glycerophospholipids and steroids. These lipids are used for structuring membranes as well as for displaying molecules on the membrane surfaces for signaling and molecular recognition. So, signaling में role होता है as well as recognition, right? So basically we know lipid bilayer arrangement of membrane. So we have discussed this in detail. Okay, that also comes in this section, but I am not discussing that because we have discussed that in cell biology in much detail, right? I will attach that video. Please watch that video. Okay, that is also very important. Lipids have uses in the cell beyond energy storage, right? Not only energy storage is a function. Beyond that also there are many uses and construction of membranes. What are those uses? Many lipids are present in the cell at smaller amounts, right? Smaller amounts में present है than those making up membrane or being stored as fat. These lipids can function as cellular messengers, right? Messengers function as hormones also, electron carriers or pigments. Okay, steroid hormones. We know this is a fat. The chemical properties of lipids are related to their structure and composition. As in studies of other biomolecules, methods such as enzymatic, chromatographic, and mass spectrometry, this is basically techniques can also be used to identify lipids, okay, identify and determine their atomic structure. So techniques are important for determination, identification of biomolecules, all the biomolecules, whether it be carbohydrates, fats, proteins, right? This is a table of naturally occurring fatty acids, structure, properties, and nomenclature. Okay, so you can check here. This is a carbon skeleton, structure, systematic name, common name, right? melting point. Okay, and basically this is the structure you can see. Here you can see no double bond. So these are saturated fatty acids. And when double bond is present, these are unsaturated, right? And you can see our position of our bond is written with carbon number and double bond present. Okay, 1, 1, then here 2, 3, and here in the bracket, positions of bonds are mentioned. You can check likewise, as well as the numbers, names, systematic name, common name. Okay, just a rough idea. This is one more table where you can see structural formulas of some carbon 18 fatty acids. Okay, just arrangement is different. Stearic acid, folic acid, linoleic acid, and alpha linoleic acid. One more table, same saturated fatty acids, you can see, and unsaturated fatty acids. All double bonds are cis, right? So basically, see, omega fatty acids we know, omega fatty acids, omega 3 and omega 6. So the carbon which is near to this carboxylic group is known as alpha carbon. Okay, you can see here, this is the alpha carbon. कार्बन जो कार्बोक्सिलिक ग्रुप के नजदीक है और जो लास्ट में है मिथाइल वन दैट इज नोन एज ओमेगा के ओमेगा एंड फ्रॉम दैट ओमेगा कार्बन द प्रेजेंस ऑफ डबल बॉन्ड सी हियर between the 3 and 4 carbon, so this is a omega 3 fatty acid. Okay, omega 3 fatty acid, and when the double bond is present in between 6 and 7, it is omega 6 fatty acid, right? So this is about polyunsaturated fatty acid which are important, right? Let's move further. So here you can see this we have studied in membrane lipids, right? Very important property. Difference between a linear chain, saturated and unsaturated, basically. So when the double bond is present in unsaturated fatty acid, it gives a band shape. Okay. A band shape means it is known as king. Produces a king. Okay. We can say band in simple words, right? Band because of unsaturation. And यहाँ पे saturated है, linear है chain, so there is no band. So you can see when there is saturated fatty acid, we can see arrangement is like a stable है arrangement. This we can call stable arrangement. And when there is unsaturated fatty acids, there is instability, unstable arrangement. Okay, now this is mixture of saturated and unsaturated fatty acids. So this is causing instability. Okay, and this is stable, a stable condition. Now this is a mixed triacylglycerol. Okay, this is basically ester of fatty acids and glycerol. In this case, all the three fatty acids are different. Okay, and when the three fatty acids are different, this the chiral carbon, right? This shown in the red one is the chiral carbon. You can see it is written when glycerol has different fatty acids at C1 and C3 position, C2 is the chiral carbon. This is the adipose tissue. Adipose tissue में fat storage होता है, right? You can see this white granules where fats are deposited as a energy storage. Now, natural fats at 25 degrees Celsius. Fatty acid composition of 3 food fats. Okay, now see different types are there: liquid, soft solid, and hard solid. Okay, soft solid means jelly, like we can say, तो लिक्विड में ऑलिव ऑयल है, ऑलिव ऑयल। नाउ व्हाट इज दिस केस? The unsaturation. Unsaturation means double bond presence, right? Unsaturation is giving instability. So when unsaturation is present, this will lead to liquid what we can say liquid fats. Okay, liquid present होगा. And when saturation is present, only saturation, then it is solid. अभी इसमें भी टाइप है based on chain length. Okay, chain length. When there is long chain, it will give rise to hard solid. Okay, and when chain is small, this will give rise to soft solid. Okay, this is a soft solid. You can see this butter is a soft solid and beef fat is hard solid, right? So इसमें green color that is for unsaturation present in high amount. Here you can see almost equal unsaturated, saturated, right? Carbon 618, 16, 16, C18 and C4 to 14 saturated. Now in this case hard solid beef fat can see this is present in high amount, saturated one, right? So I told you, you have to just remember this. Unsaturation will give rise to liquid fats, instability condition. Saturation will give rise to solid condition and इसमें भी small chain अगर है soft solid, long chain अगर है तो hard solid. Again, again, uh, one more thing to study is about the melting temperature. So unsaturation, saturation. Okay, saturation I will write small. Small means small chain. To saturation, long chain. Melting temperature will increase. Okay, melting temperature increases. Right? So let's erase this so that it is easy to read. See, olive oil, butter, and beef fat consist of mixtures of triacylglycerol differing in the fatty acid composition. The melting point of these fats and hence their physical state at room temperature 25 degrees Celsius are a direct function of their fatty acid composition. Olive oil has a high proportion of long chain unsaturated fatty acids. Okay, olive oil has high proportion of unsaturated fatty acid. हमने देखा अभी which accounts for its liquid state at 25 degrees Celsius. तो room temperature पर liquid है because of unsaturated fatty acids. High proportion. Now higher proportion of long chain saturated fatty acids. Saturated fatty acids in the high proportion in butter increases its melting temperature. Okay, melting point. So butter is soft solid at room temperature. Okay, beef fat which has even higher proportion of long chain saturated fatty acid is a hard solid, right? So as the proportion of saturated fatty acid increases, solidity increases as well as melting point increases. This is the biological wax, triacontan oil, palmitate. major component of beeswax. Okay, mixture of palmitic acid with alcohol triacotanol. So again, this is a ester. The beeswax of honeycomb is firm at 25 degrees Celsius. So 25 degrees Celsius is the room temperature, completely impervious to water. Okay, impervious to water means water cannot enter. It does not allow water to enter. The term wax originates in the Old English "viox", meaning the material of the honeycomb. Okay, just idea. This is like a summary. Lipids are water-insoluble cellular components of diverse structure that can be extracted from tissue by non-polar solvents. Non-polar solvents use करके हम extract कर सकते हैं. Almost all fatty acids, the hydrocarbon component of many lipids, have an even number of carbon atoms. Okay, even number of carbon atoms present, usually 12 to 24. They are either saturated or unsaturated, with a double bond almost always in the cis configuration. Okay, triacylglycerols contain three fatty acid molecules esterified to the three hydroxyl groups of glycerol, as we saw example of mixed triacylglycerol. Simple triacylglycerol contain only one type of fatty acid. Okay, simple में only one type of fatty acid होता है, and mixed triacylglycerol में two or three types of fatty acid are present, where C2 is the chiral center in the mixed triacylglycerol. Triacylglycerols are present in many foods, right? And are primarily used for storing energy storage, and providing insulation in animals, means acting as protection against cold weather. Lipases is a enzyme which degrade lipids, okay, or break down the fatty acids, release fatty acids from storage so they can be used as fuel, right? Since many natural fats can easily turn rancid, okay, rancid turn होते हैं, partial hydrogenation has been used to extend their shelf life, right? This is very important. So rancidity prevention is done by partial hydrogenation, and this is for extend the shelf life of fats. This process can produce trans fatty acids. Okay, पहले cis fatty acids होते हैं. Okay, cis double bond we saw, almost always cis double bond is present. After partial hydrogenation, these are converted to trans fatty acids. So basically these are harmful. Okay, these are harmful, causing heart diseases. So trans fatty acid increase the risk of coronary heart disease. As a result, their use in prepared and processed food has become highly regulated. Okay, waxes are esters of long chain fatty acids and long chain alcohols with diverse uses in biology and industry. Right, so this is just a summary. Now next, so this is a phospholipase which degrade the phospholipid. There are types: phospholipase A1, phospholipase A2, C, and D. This molecule is basically PIP2, phosphatidylinositol 4,5 bisphosphate. This is used in the signaling process, right? Has a role in signaling. So phospholipase A1 and A2 hydrolyze the ester bond of intact glycerophospholipid at C1 and C2 of glycerol, right? Phospholipase A1, A2, C1 and C2 का bond hydrolyze करता है, ester bond. When one of the fatty acid has been removed by a phospholipase, the second fatty acid is removed by a lysophospholipase, which is not shown, right? So there is one more phospholipase in the action. lysophospholipase. Phospholipases C and D each split one of the phosphodiester bond in the head group. Some phospholipases act on only one type of glycerophospholipid, such as phosphatidylinositol 4,5-bisphosphate shown here or phosphatidylcholine. Others are less specific. So the information which is given here and these pictures are very important actually. Questions are asked directly from this. Basically, this is related to membrane lipids. Okay, first you have to know the membrane lipids, which we discussed previously. I will attach that video in the I button. Please watch that. Okay, and then continue from here. Okay. So here is a pathway for breakdown of GM1, globicide and sphingomyelin to ceramide. Now, a defect in the enzyme hydrolyzing a particular step is indicated by this sign. Okay, cross sign. The disease that results from accumulation of the partial breakdown produces is noted. Now see, there are enzymes present, right, to break down the lipids, sphingolipids. Here enzyme is there, and when this enzyme is absent, this disease is caused. Okay, same here this one. This is very important. This enzyme, hexosaminidase A. Okay, hexosaminidase A. When this is absent, it produces THS disease, right? THS disease. Then this you have to remember. This is asked in exams. Then again, these are the enzymes given. So what happens basically in the disease? If we say there is no complete metabolization or breakdown seen. So, abhi suppose this is a disease, right? Gaucher disease. Okay, Gaucher disease. Now in this disease, what will happen? This product will not form. Ceramide बनेगा नहीं. तो यहाँ तक accumulation होगा. This product will be accumulated in the cells. Okay, and this can cause toxicity. So it will be any disorder if you say. Okay, मतलब पूरा breakdown होना important होता है. अगर वो नहीं हुआ तो फिर जो पहले वाला product है that will get accumulated in the cells and that will give rise to toxicity. Okay, toxicity or harmful effects will be produced. So I will erase this so that this is clear actually. You can see here these are more disorders given of enzyme. Now this is sphingomyelin, phosphocholine is separated, ceramide is separated. Okay, now this enzyme is sphingomyelinase, right? And when this is absent, it gives rise to Niemann-Pick disease. This is also important disease, right? You remember this name, sphingomyelinase enzyme deficient in Niemann-Pick disease. So this is also like a summary points about membrane lipids, right? About membrane lipids. Lipids with polar head and non-polar tail are major components of membrane. The most abundant are glycerophospholipid, which contain fatty acid esterified to 2-hydroxyl of glycerol. The third hydroxyl of glycerol is esterified with the polar head group via phosphodiester bond. So glycerophospholipid में 2-hydroxyl group के साथ esterification, right? And the third hydroxyl is associated with esterification से attach है polar head group via phosphodiester bond. So common glycerophospholipids are phosphatidylethanolamine, phosphatidylcholine. Differ in the structure of the head group. Okay, you will see the structure in the separate video, right? Heart tissue, plasmalogen and platelet activating factor are examples of glycerophospholipid containing ether-linked acyl chain. Okay, ether-linked acyl chains होता है. Chloroplast membranes are rich in galactolipids, right? Composed of diacylglycerol with 1 or 2 linked galactose residue and sulfolipids, diacylglycerol with a linked sulfonated sugar residue. Okay, sulfolipid means sulfonated sugar is present. Some archaea have unique membrane lipids with long chain alkyl groups ether linked to glycerol. Okay, archaea में ether linkage. These lipids are stable under harsh conditions. to which these archaea live. So this gives them stability. Thus sphingolipids consist sphingosine instead of glycerol. Right? Sphingosine present होता है. The three subclasses of sphingolipid are ceramide derivatives, sphingomyelins, neutral glycolipids, and gangliosides. Sphingomyelin have cooling hair groups. Other classes have sugar component. Okay. Sphingolipids are abundant in plasma membrane of neurons. Sphingomyelin, right? Neurons में present होता है, and they define human blood groups also. Okay, human blood group में भी sphingolipids present होते हैं. Phospholipases degrade glycerophospholipid as we saw and catalyze hydrolysis at specific position within the structure. Okay, hydrolysis करते हैं lipids का. Steroids have 4 fused rings. Okay, and the hydroxyl group. We know cholesterol, which is a major sterol found in animal, is both a structural component of membrane as well as precursor to variety of steroids. Right, variety of steroid का precursor भी है. Example steroid hormone, estrogen, progesterone, estradiol, testosterone. Right. Now we are starting with next section, that is phosphatidylinositol and its derivatives. You can see here this is the phosphatidylinositol molecule. So in this what happened, the glycerol phospholipid is attached at C1. Right, you can see this is a C1. इस पे glycerol molecule attached है. Phosphorylation of the remaining inositol hydroxyl group forms signaling molecules such as phosphatidylinositol 4,5 bisphosphate. Okay, PIP2, inositol 1,4,5 bisphosphate, and phosphatidylinositol 3,4,5 triphosphate, PIP3. So different signaling molecules are produced by phosphorylation at other positions. But glycerol is attached to this C1. Okay, a useful trick for remembering the inositol carbon numbering. मतलब आप remember कैसे कर सकते हो, by giving this similarity to the turtle. Okay, to compare the chair conformation of inositol to a turtle. Begin numbering with C1 as the rigid front flipper and move counterclockwise around the structure. With C2 the head, C5 the tail. You can see this, just nothing. See, this is a C1 where glycerol is attached, right? And rest other position you can see, this is the flipper, this is the tail, 2 and 5 position respectively. So you can see these are the arachidonic acid and some eicosanoid derivatives. So arachidonic acid is a precursor of eicosanoids, including the prostaglandins, thromboxane and leukotrienes, lipoxins. तो इसका precursor है arachidonic acid. So in prostaglandin E2, C8 and C12 of arachidonate are joined to form characteristic 5-membered ring. In thromboxane A2, right, the C8 and C12 are joined and an oxygen atom is added to form the 6-membered ring. Now, non-steroid anti-inflammatory drugs, basically painkillers, okay, such as aspirin and ibuprofen, block the formation of prostaglandin. So, prostaglandin, now see, these are produced in inflammatory cases. Okay, inflammation when there is infection. So now ये जो पेन किलर्स है एस्पिरिन आइबुप्रोफेन ब्लॉक करते हैं फॉर्मेशन ऑफ प्रोस्टाग्लैंडिन्स एंड थ्रोम्बोक्सिन obviously from arachidonic, okay, by inhibiting the enzyme COX, right? COX, I will write here COX is what? That is cyclooxygenase, right? Cyclooxygenase, prostaglandin H2 synthase. Leukotriene A1 has a series of 3 conjugated double bonds and no cyclic moiety. So 3 double bonds are present. There is no cyclic moiety. Lipoxygens are also non-cyclic derivative. Lipoxygen non-cyclic derivative arachidonic with several hydroxyl groups. Got it? See, this is a steroids derived from cholesterol. You can see testosterone. So these are hormones. Male sex hormone produced in testes. Cortisol hormone produced in the adrenal cortex regulate glucose metabolism. Prednisone, okay, prednisone. This is a synthetic steroid used an anti-inflammatory agent. So basically, this is used as medicine. Synthetic means man-made. Theta estradiol. This is female sex hormone produced in the ovaries and placenta. Then aldosterone. Basically, this hormone is produced in adrenal cortex. Regulation salt excretion. Then this is brasinolid. This is in plants. Brasinosteroid, growth regulator found in vascular plants. So you can see fused ring structure is present everywhere, which is common in the steroid, right? Steroid fused ring structure present everywhere in this molecule. So these are derived from cholesterol. So this is vitamin D3 production and metabolism. Now before we go further, I will tell you vitamins A, D, E, K. These are fat soluble. So obviously these are related to fats, right? Okay, hydrophobic है. Okay, fat soluble है. So these are related to fats. We have to remember this. So cholecalciferol, vitamin D3 is produced in the skin by UV radiation of 7-dehydrocholesterol, which breaks the bond shaded light red. See, this bond is broken. In the liver, a hydroxyl group is added at C25. Okay, C25 hydroxyl group is added. In the kidney, a second hydroxylation at C1 position produces the active hormone 1-alpha 25-dihydroxy vitamin D3. These hormones regulate the metabolism of calcium in kidney, intestine and bone. Dietary vitamin D prevents rickets. Rickets prevents vitamin D, a disease once common in cold climates where heavy clothing blocks the UV component of sunlight necessary for the production of vitamin D3. Cold weather में common होता था. Why? Because using heavy clothing, sunlight cannot penetrate within the bones, right? Which is important for the production of vitamin D3. Rickets result in weak or soft bones in children. It can be often identified by the bowed legs and often other bone deformities. So you can see this picture. The first molecule 7-dehydrocholesterol. Because of UV, the bond is broken. Okay. Then in the liver, hydroxylation takes place. First hydroxylation at C25 position and second hydroxylation in the kidney at C1 position. Okay, and this is the active hormone 1-alpha 25-dihydro vitamin D3. And this is again the beta carotene vitamin A1 as precursor of retinoids. So we know this is having function in the vision, right? So you can read this. Okay, keratin enriched rice. You can see here. Basically, this rice is a golden rice. Okay, which is produced by genetic engineering, right? So in this, the beta carotene is produced. Okay, beta carotene is produced, which gives this rice a yellow color, and this can treat the vitamin E deficiency. Basically, not treat, prevent vitamin A deficiency and its tragic health consequences. Okay, so you can see this is a wild type and this is a genetically engineered rice. Okay, so your keratin is produced by genetic engineering, and this comes under basically biotechnology section, techniques part, but this is important, so I added here. Next is isoprene compound. You can see this isoprene is a 5-carbon compound राइट. एंड सपोज हाउ समटाइम द क्वेश्चन कम्स, ऐसे पूछते हैं कि कितने कार्बन प्रेजेंट है. हाउ बेसिकली सपोज 10 यूनिट्स आर प्रेजेंट of isoprene, 10 units are present, then this is equal to 50 carbon, right? So you can see इसे हम काउंट कर सकते हैं by multiplication because we know the isoprene has 5 carbon. Now you can see here some other biological active isoprene compound or derivative, right? And you can see these dashed lines are basically separating the isoprene units. Okay. Now from this, what is important? Ubiquinone. Okay, in most mammalian tissues, also called coenzyme Q, that is OQ, has 10 isoprene units. Okay, 10 isoprene units present है. Dolichols of animals have 17 to 21 isoprene units, 85 to 105 carbon atoms. Bacterial dolichols have 11 units, and dolichols in plants and fungi have 14 to 24. Right. So here different compounds are there. Vitamin E, this is an antioxidant. Vitamin K, blood clotting cofactor. Warfarin is an anticoagulant. Ubiquinone, electron carrier, as well as plastoquinone, electron carrier है. Dolicol is a sugar carrier, right? Next, lipids as pigments in plants and bird feathers. Okay, compounds with long conjugated system absorb light. So how light is produced? Light is produced by absorption of light in the visible region of the spectrum. Now there are differences in the chemistry. मतलब chemistry में differences है of these compounds which produces pigment of strikingly different colors. And how the birds acquire this pigment? Basically by eating plant material that contain carotenoid pigments. Okay, so carotenoid basically pigment produce करते हैं, color produce करते हैं. So that gives that color, the feathers color, red or yellow. Right, specific color production takes place, such as canthaxanthine and zeaxanthine. These are the two compounds. You can see from the diagram, canthaxanthine produces bright red color. You can see this diagram, and zeaxanthine produces bright yellow color. Okay, the differences in pigmentation between male and female birds are the result of differences in intestinal uptake. अभी male and female में differences है. How? Why so? Because there are differences in the uptake as well as processing of carotenoids. Got it? Now these are the three polyketide natural products used in human medicine. So basically, what are polyketides? Secondary metabolites. Right, secondary metabolites है. Okay, so your example is erythromycin, which is antibiotic against the bacteria. Amphotericin B means antifungal, means against the fungal infection, and leostatin, that is statin, basically for treating the heart diseases. Right, when the cholesterol is high, statins are used to prevent heart complications. This is again a summary. Some types of lipids, although present in relatively small quantities में present होते हैं, play critical role as cofactor or signal. We saw this membrane sphingolipid and derivatives of phosphatidylinositol such as PIP2, PIP3 are used as signaling molecule or for nucleating formation of multi-protein complexes. Prostaglandin, thromboxane, leukotriene and lipoxin, all of which are eicosanoid derived from arachidonic, okay, are extremely potent hormones involved in reproduction, inflammation, regulating blood pressure and other bodily processes. Right, so these are also hormones which are involved in different functions. Now, non-steroidal anti-inflammatory drugs, basically painkillers, inhibit formation of some prostaglandin and thromboxane. Inhibit करते formation basically by inhibiting the enzyme cyclooxygenase. Steroid hormones such as sex hormone are derived from sterols. They serve as powerful biological signals, right, and move through the bloodstream to alter gene expression in the target cell. मतलब blood stream से move करके target cell के पास जाते हैं. And what do they do there? alter the gene expression, right? So plants produce volatile lipids to attract or repel other organisms, attract करने के लिए और repel करने के लिए, as well as for communication purpose. Many of these lipids are used as fragrances in perfume. Vitamin A, D, K are fat soluble compound, right? मैंने बताया ये फैट से रिलेटेड है, A, D, K fat soluble. These are derived from isoprene units and all play essential role in the metabolism or physiology of animals. Vitamin D is a precursor to hormone that regulate calcium metabolism. तो विटामिन डी का रोल है कैल्शियम मेटाबॉलिज्म में. विटामिन ए का रोल है विजुअल पिगमेंट ऑफ द वर्टिब्रेट आई, रेगुलेटर ऑफ जीन एक्सप्रेशन ड्यूरिंग एपिथेलियल सेल ग्रोथ. गॉट इट. विटामिन ई एंड के and quinone can be oxidized or reduced by cells. मतलब सेल्स ऑक्सीडेशन रिडक्शन कर सकते हैं. तो विटामिन ई का फंक्शन क्या है? प्रोटेक्शन ऑफ मेम्ब्रेन लिपिड, राइट? मेम्ब्रेन लिपिड का प्रोटेक्शन फ्रॉम ऑक्सीडेटिव डैमेज. एंड विटामिन के इज एसेंशियल फॉर ब्लड क्लॉटिंग. देन क्विनोन स्पेशली एसेंशियल फॉर कैरिंग इलेक्ट्रॉन्स. हमने देखा है यूबी क्विनोन जो प्रेजेंट होता है इलेक्ट्रॉन ट्रांसपोर्ट चेन में, राइट? टू ट्रांसफर इलेक्ट्रॉन, okay, carry electron during the reactions that drive ATP synthesis. तो ATP synthesis का फॉर्मेशन होता है इन माइटोकॉन्ड्रिया एंड क्लोरोप्लास्ट. देन डॉलिकॉल्स एक्टिवेट एंड एंकर शुगर, राइट? शुगर का एक्टिवेशन करते हैं, एंकर करते हैं. एंकर मीन्स अटैचमेंट, ओके, टू सेल्युलर मेम्ब्रेन. So as I said, this is called glycosylation and we have discussed this in the cell biology. The sugar groups are then used in the synthesis of complex carbohydrates. Complex carbohydrate का formation होता है glycolipids as well as glycoprotein. Then lipidic conjugated dienes. Okay, lipidic conjugated dienes means these are conjugated. Okay, conjugated means addition, integrated, right? Serve as pigments in flowers and fruits, right? Flowers and fruits में होते हैं and give bird feathers their striking colors. मतलब different colors we see in the fruits, flowers, feathers of birds. These are because of these compounds. Polyketides are natural products widely used in medicine. Okay. Then this is the next table. Important. You can see 8 major category of biological lipids. Fatty acid. Examples are oleate, pyrrole CoA, palmitoyl carnitine. Glycerolipids, diacyl and triacylglycerol. Glycerophospholipids, phosphatidylcholine, phosphatidylserine, phosphatidylethanolamin, phosphatidylinositol, right? Sphingolipid, sphingomyelin, ganglioside GM2. Pyrrolipids, cholesterol, progesterone, bile acids. Phenolipids, farnesol, geranol, retinol, ubiquinol. Renol basically derived from isoprene, right? Saccharolipids, example lipopolysaccharide. Polyketides, example tetracycline, erythromycin, aflatoxin B1. Now this is in relation with techniques. Okay, techniques part with lipids. Okay, means lipid के लिए हम techniques को कैसे use कर सकते हैं. So basically techniques will be discussing in the separate section, right? A separate playlist. So basically यहाँ पर हम सिर्फ लिपिड पॉइंट ऑफ व्यू से देखेंगे व्हाट इज इम्पोर्टेंट. So in determination of lipid composition, lipid can first be extracted. पहला लिपिड का एक्सट्रैक्शन करना है फॉर डिटर्मिनेशन पर्पज फ्रॉम टिश्यूज विथ ऑर्गेनिक सॉल्वेंट्स. ओके, ऑर्गेनिक सॉल्वेंट को यूज करके. लिपिड्स इन मिक्सचर्स कैन बी सेपरेटेड. सेपरेशन करने के लिए इम्पोर्टेंट है polarity, right? On the basis of the polarity and interaction with polar materials such as silica using adsorption chromatography methods such as SPLC or TLC. तो chromatography separation के लिए important होता है, right? Mixtures को separate करने के लिए. Same with lipids. GC, that is gas chromatography, volatilizes lipid so that they can be carried by stream of inert gas and resolve based on the ability to partition into a soluble column material. Phospholipase is specific for one of the bond in a phospholipid can be used to generate simpler compounds for subsequent analysis. ओके, अब इस फॉस्फोलिपेजेस जो स्पेसिफिक है उनको हम यूज़ कर सकते हैं. वाई सो? इसलिए लॉन्ग चेन फॉस्फोलिपिड है, तो इसे हम शॉर्ट चेन में कन्वर्ट कर सकते हैं बाय फॉस्फोलिपेज. हाई रिजोल्यूशन मास स्पेक्ट्रोमेट्री अलाउ एनालिसिस ऑफ क्रूड मिक्सचर्स. क्रूड मिक्सचर्स का एनालिसिस पॉसिबल है विदाउट रिफ्रैक्शनेशन. दिस इज कॉल्ड शॉटगन अप्रोच. शॉटगन अप्रोच. लिपिडोमिक्स combine powerful analytical technique to determine the full complement of lipids in a cell or tissue. right? मतलब एक सेल में टोटल लिपिड कंटेंट डिटरमाइन करने के लिए लिपिडोमिक यूज कर सकते हैं. तो बेसिकली जो फुल कंटेंट होता है ऑफ लिपिड दैट इज कॉल्ड लिपिडोम इन अ सेल और अ टिश्यू. एज वेल एज वी कैन assemble annotated databases that allow comparison between lipids of different cell types as well as under different conditions. Means what? Suppose this is the liver cell, इसका हम लिपिड कंटेंट डिटरमाइन कर सकते हैं एंड देन वी कैन कंपेयर दैट द किडनी सेल. मतलब यहाँ कौन से लिपिड प्रेजेंट है एंड यहाँ कौन से लिपिड प्रेजेंट है अंडर डिफरेंट कंडीशंस ऑल्सो. मीन्स relating that with the metabolism. Okay, we can do that. So this was all about lipid. I have highlighted the important things. Membrane lipids हमने separate वीडियो में डिस्कस किया है. Watch that video. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to our channel, and stay tuned for the upcoming videos. Thank you.